So the things that you need to know from this presentation, the 3 stages of cell communication. So that would be reception, transduction, and response. Okay. You need to know how G protein coupled receptors, okay, how they receive signals and start transduction, how receptor tyrosine kinase receives cell signals and start transduction, how cell signal is amplified by a phosphorylation cascade, how a cell response in the nucleus turns on genes while in the cytoplasm activates an enzyme, and then what apoptosis means and why it's important to normal functioning of multicellular organisms. And we've talked about apoptosis a little bit before. So signaling, the term signaling is basically the entire process from reception through a response and something happening. So the actual signal being released is part of a response from another cell. And then we have our other stages. So we'll kind of go through that. Now, animal cells communicate through direct contact, secreting local regulators, and long distance. Now, what that means, so we have, it actually happens with plants too, just a little different because our cells are a little bit different. So we have these junctions here, and we can actually have one cell talk to another. When I say talk, I'm just meaning signaling. So we can have them signal each other by sending a molecule that it makes through there, the gap junction into the other cell. And remember that our plants have that plasmodesmata through the cell wall, kind of like little tunnels, to be able to allow us to do this. Now, cell-to-cell recognition, seeing another cell and being like, yeah, you're cool, you're one of us, stay here, that is considered a type of cell signaling. Having your body go, oh, wait, you're not one of us, you need to go away, we're gonna kill you. That's a type of cell signaling. So all of these things are ways that our body allows us to do things like growth. Most of our homeostasis are maintaining a stable internal environment. It comes from signals. And then we can also recognize things from the outside world. When food is entered into our stomach, we have signals that are sent to start making our stomach actually squish everything up, peristalsis, all kinds of things. So here we have our very close signaling. We have the signal molecules released. Okay, signal molecules are released and then target cells receive them. Okay, here we have, this is 2 different neurons. Neurons are nerve cells. We're going to talk about the neurons a lot in this presentation. Okay, so this is the end of one neuron and this is the next one. Now think of your nervous system like railroad tracks. Okay, and we have to send the signal along the entire length of that track. So we have to relay the messages all the way down. And this neuron releases a signal to tell this neuron to continue the message. Okay, we call these neurotransmitters. For long distance signaling, we can have a cell, here's an endocrine cell, releases a signal into the bloodstream and it goes to the target cell and has whatever effect take place. Now, this could be adrenaline. So we release adrenaline into the bloodstream. It goes to the cells within your heart, and then it is signaling to make the contraction happen faster. Okay, so different signals produce different responses. The 3 stages of signaling include reception. So we receive a signal, and that signal molecule that we've been talking about, we call that a ligand. Okay, it has a specific name, a ligand. That means that we can talk about a signal molecule and not have to like say as many words to talk about what exactly we're talking about. So in science, we call it a ligand. We know that means it's a signal molecule that is being produced and sent out for a reception. We have transduction, which is like sending the signal, relaying it within the cell, sending it to where it needs to be. And then we have response, and response is where the cell actually does something with that signal. What does it want me to do? What is it telling me? What are the instructions? Okay. So reception, we have proteins on our membranes, and we've talked about those a lot. And these proteins are specific. Okay, they receive our ligand, our signal molecule. And again, they're specific. So this protein receives this signal. That's reception. These two coming together, that's all it is. So we've received the message. Okay. And again, it is very specific, very specific. So if I have a cell, let's say this is, let's say it's growth hormone. Okay. Now growth hormone, we want it to trigger in like pretty much all of our cells, not every single cell, but pretty much all of them. So all of our cells will have this. But if you think about, let's see, estrogen and testosterone are sex hormones. We don't necessarily want every cell on our body to suddenly start sprouting hair. Okay, so not every cell has a receptor for the hormone that says it's time to grow hair. Okay, so not every cell has all of the same receptors. We have plasma membrane receptors. So that's what we were looking at on the previous slide. And these are for water soluble ligands, okay, water soluble. And then we have intracellular receptors. That means it's in the cytoplasm. And these are for hydrophobic and small ligands, things that can sneak through and don't need a protein to help get into the cell. Testosterone and nitric oxide, those are examples. Again, remember we talked about shape and function. So when the ligand binds to the receptor protein, the protein changes shape. Remember how our enzymes could change shape? Same thing happens here. And by that change of shape, that is what initiates our signal transduction. Okay, so for signal transduction, we are going to look at, we have things like this is a segment of a signal molecule. Okay, this receives a signal, and then this is going to interact with a G protein. Okay, so it has to do with the shape change here. Whoa, 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 whoa. To go here. Okay, I just had a couple of you ask me to slow down. I'm sorry, guys. I forget when I can't see like all your faces looking at me, I tend to forget that I'm going too fast. All right, so let me go back. I'm going to review a little bit because I think I went too fast. So let's go back to reception. So here we have reception. We have our signal molecule. This molecule we call a ligand. It is received by our receptor protein. So our receptor protein is attached to our signal molecule, our ligand. When the ligand and receptor come together, this is called reception. It's a pretty simple thing there. Now, our receptor molecules are specific, just like our enzymes were, and they have a shape change that happens, just like with enzymes. So when our signal molecule attaches, okay, our ligand attaches, it causes a shape change here. And that change in shape is going to be what tells everything else to relay the message, because this protein has to stay here. Okay, the protein has to stay within the membrane. That's where it lives. But we need to get that message to the part of the cell to have a response. So again, we're specific. We have two types. They can be on the membrane, or it can actually be floating within the cytoplasm. These are intracellular receptors. And the same thing happens as our plasma membrane receptors. It's just that the molecule finds it inside the cell instead of latches on the membrane. Okay, so here's an example of that protein receptor on the membrane. So here's one of our plasma membrane receptors. Okay, it has a binding site. That means the ligand is going to land right here. When the ligand lands here, it allows this part to change shape and send its signal. Okay, so our receptor is really just a relay point. Now, if we look at G protein coupled receptor, okay, here is our protein. It is a couple, so it is a protein that is coupled with that G protein. Now, couple means two. So here's our membrane protein, here's our G protein. When it is activated, it changes shape and it like pulls us back into it. Imagine like a bow and arrow. And when I put in my ligand, it pulls this protein back to it. So we wind up with it something like this. And this seems really weird, but it is so cool. What happens when they do this is we use some energy. GTP is a lot like ATP. Okay, it's an energy molecule that we don't need to know too much about, but know that it's energy. And it actually shoots it out. And when it shoots it, it's going to hit this enzyme. Okay, so it pulls it back and then it shoots it out. And then when it shoots out, it hits this enzyme, and this enzyme now sends a reaction and it releases this so it can go back to do its job again. Okay, so we've received a message and the enzyme now has to do something with it. Okay, G protein coupled receptor, it's a couple of proteins. It pulls it back like a bow and arrow and shoots it and hits the other protein. What happens then is this protein starts a cascade of reactions. So this protein changing shape, this hits it, causes it to change shape. So it's like a chain reaction. It means that this is going to do its thing, which causes this to do its thing, which causes a response. So it winds up being this whole chain of different things happening. It's like a relay race. You hand it over to the next thing, which takes it to the next thing and takes it to the next thing. And we wind up eventually with a response happening. Okay. Now I want you to take a look here at the right-hand picture. We have our G-coupled protein. It causes this to do its job. This message is sent over, but now this protein, which received the message, doesn't let just one in. It can let a whole bunch do its job. And now instead of one signal, I have all these different messengers that can go make a response happen. Okay, so it's a chain reaction, but sometimes that chain can trigger a whole bunch. So it's like if I made a phone call to Tim, and then Tim called John, John called Gabriella, Gabriella calls Fiona, and then Fiona calls Javier, but Javier doesn't call one people. He's like calling everybody. Okay, and so it gives the message to everybody. Okay, now it can spread even faster because instead of 1 to 1 to 1 to 1, now I have 1 to like 50, and then all those 50 can go spread the message. Okay, so we can increase the response when we have these cascades. Okay. It's just like a domino, yes, like a domino effect, and like spreading rumors, definitely. Okay, so it starts as a domino, but have you ever seen in videos that it's not, sometimes they can trigger more than one? A domino falls and it'll hit 2 or 3 and it starts spreading out. That's what this is like. Okay, now we have signals like this happening inside our cells all the time. It just depends on what the signal is. So like right now, me talking, there's thousands upon thousands of signals being sent every second within my body because I have to get the information from my brain. I have to get it to my mouth, tell my mouth how to move, how to get the air to come up through my lungs, how to change my mouth so that the air coming past my vocal cords makes the sounds I want. My hands are moving all over the place. My head's moving. So thousands of signals being sent. Okay, and that's just for movement. That doesn't even include things like breathing is a message that's being sent through your bloodstream. Okay, and that bloodstream message is going to the brain, which is then telling the lungs to move and all these different things happening. So depending on what signal we're talking about, it's happening potentially thousands or millions of times in your cells every second. Okay, now there are some that don't happen very often. Puberty is a whole bunch of signals being sent to your body to change, but it doesn't happen constantly. Okay, imagine if it started when you were a baby. Can you imagine toddlers walking around with, big buff toddlers with hairy armpits and, that would just be like creepy. Imagine their voices going all deep, or, imagine girl toddlers wearing bras. That's just not okay. So we have some signals that we want to have happen regularly. Other signals are very regimented in their times. And we do that through having specificity in when signals are received and what they can do. Okay, so we don't start producing all those puberty hormones until there are signals in the brain that are sent to the cells. So we have part of the brain that works between the nervous system and the message system, the endocrine system. It's like, it links the two systems together. And so our nervous system is predominantly electrical. Okay, we'll talk about that a little bit more. Whereas the endocrine is all chemical. And we have to let them talk sometimes. So the message between those and when we send those and how we send those is what regulates when we can do all this. The G, what does the G mean? I don't know. I'll be honest. I don't know what the G is. I'm guessing it's a type of protein, but let me look into that. I'm sorry, I don't know that answer. G protein. To me, my brain, it's always just been a name. It's the G protein. I'm Mrs. Langley, it's the G protein. So I will look into that, I'm sorry. Let's see, is there an example from the plasma membrane? Oh, this is an example, yes. So this is an example of a plasma membrane protein that is receiving our ligand and sending the signal. Now, the only difference is this ligand is big. It can't get through the membrane, so we have to have a protein on the outside, just like we talked about when we talked about movement into and out of the cells. If we were looking at a cytoplasmic membrane, or I'm sorry, cytoplasmic protein, it just means this ligand would be a tiny ligand, and it could slip through here, and this protein would be floating inside our cell instead. But then the messages and all this happens exactly the same. Okay, so imagine we just take this and pull it into the cell, and we have a little ligand that can slip through and get over to our protein. Make sure I got all the questions here. All right. Now, another type of receptor is called a tyrosine kinase receptor. And these are pretty cool because it allows us to not waste energy. If we have a ligand in the system, so let's say this is in the bloodstream, and we have a little bit of a ligand in there, a little bit of a signal, a little bit of a hormone, it's not like every cell freaks out and has to send signals. With the tyrosine kinase receptors, we have two separate ones, and they wait, and if they both get a signal, they'll come together and allow it to do its job. This allows so that, like, let's say one gets it, but there's not enough in our bloodstream for the other to get it. It's like, why am I going to waste the energy when this is probably just a false alarm? Okay, so it allows us to be very specific and make sure we really want this signal sent. So one receives it, the other receives it, they come together, and when they bind together, it allows our cascade of reaction to occur. Okay, so tyrosine kinase is two different proteins that both have to receive a signal, and then they click together and send the signal into the cell. I'm gonna wait a minute here because I know you guys, some of you are writing like crazy. So why would we want this? Question was asked, why did they do this? What would the signal be for? So there are signals that are at low levels in our body all the time. Now, because it's an easy way to think of it, let's think about our sex hormones. So there's estrogen that goes through everybody's body. Everybody has estrogen, everybody has testosterone, and they're at low levels. Okay, so now let's say a girl has low levels of estrogen in her body. Fine, whatever, we don't care. When those estrogen levels kick up, okay, so as a girl each month, there's like the cycling, the estrogen increases and decreases. So as the estrogen level increases, then this signal can be received, okay, and that might trigger the release of an egg into the fallopian tube. Okay, so we don't want the eggs to be released constantly, but this would say, okay, we know it's going to be, the estrogen's there all the time, but only when it's at high levels do we want this to happen. Okay, so this allows us to ignore when levels are too low for it to mean anything. Now these ones, I think these ones are pretty cool. Ligand gated ion channels. Ligand, we know what that is. Gated, it's a gate. We have a protein and there's a gate across the hole so that it can't just drop through. Okay, and we've seen these kinds of proteins before. It allows for passive transport. Things can just kind of drop in, flow back and forth, but we don't always want things flowing all the time. Now, this is how your nervous system works. It uses these. So these are ions, positives, negatives, things like calcium, potassium, sodium, things along those lines, okay? And if they readily moved back and forth, we couldn't create charges on either side. By blocking it, we can create a positive and negative. By opening it, we can flip that, we can change it, we can do all kinds of things. But it allows us to regulate how much goes in and out of the cell. So we have our ligand. It clicks in here, which pulls the gate open, allows things to go through, and then we release the ligand and close it again. Usually there's something that pulls this ligand out, okay? This is how neurotransmitters work, and our cells will actually look to pull back in these neurotransmitters. We'll talk about that more, I promise. So these are easy ones. They're ligand gated. So we have a gate that the ligand can open. And it's a channel or a tunnel. Okay, so that's just receiving the message. So this, does it use ATP or not? There are some that require ATP and some that don't. Generally speaking, this is passive. So there's no required. It has to do with the protein receiving, just like our enzymes did, and then changing shape. Once they've changed shape, they can pass through readily and easily. So the vast majority of these are no energy required. That's a very good question. This is just a summary of those, which you can take a look at later. Now, transduction, it's a relay system. So this gets a message, tells this molecule, which tells this molecule, which tells this molecule. This is it, just really simplified. And that's all you have to really remember, is it's sending the signal by one molecule to the next, okay, one protein to the next. Again, it's like giving a message to one person who calls one person who calls one person. Okay. Now, we can amplify this by having this one, instead of calling one person, call a bunch of people. So a cascade is a relay. So it just relays it till it gets to the target. Protein kinase is an enzyme that phosphorylates and activates proteins at the next level. And a phosphorylation cascade enhances or amplifies that signal. So this is a bunch of big words. We're going to look at proteins, okay? Protein kinase, ACE means it's an enzyme. So this is an enzyme that works with our proteins to cause phosphorylation. And we've done phosphorylation before. We did it when we did ATP synthesis. Phosphorylation means adding the protein, adding the phosphorus, okay? Don't freak out. We have reception. We've activated our relay molecule. And what happens is we're gonna start adding and taking off phosphates. And that's how we get, so we make this molecule. But to get it to go to the next one, we have to use ATP and take phosphates off, okay? It's just a way of having added regulation in here. So it doesn't just go, you know, boom, 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 boom. It has to do a little bit of work. And by having to do a little bit of work, we make sure we don't do unnecessary work, okay? So where do these phosphates come from? The phosphates within our cell, we are going to have phosphates that float around all the time because we have ATP being used in the cell. So every time ATP is used and converted to ADP, we now have a phosphate floating around in our cell. So we have to then add a phosphate and then we remove it. And then we add a phosphate and then we remove it. So it seems like a waste of time, add the phosphate, then take it back off, but it allows for specificity and it allows to make sure that we only get to the next step when we should, that we don't do it if we shouldn't be doing it. Okay. So this looks complicated. It's just that relay. Okay. But think of it like having to make that phone call. Now here's the next person and they have to make the phone call. And the next has to make the phone call. Okay. Now we can have here again, if I get this one ready, it could do the job several times and make extra phone calls. That's all it is when we are amplifying the reaction. So one message turns into a whole bunch. Okay, it's kind of the difference between like if I made a phone call versus you posted it on social media. Okay, and then somebody else posts it on social media and now it goes out to all those people as well. Okay. What regulates giving the ATP to the protein? It has to do with, so this activates this, which allows this protein to be activated. And it's basically the activation of each step that allows the ATP to be used. So there's nothing regulating the ATP use except for making this enzyme active. So when I activate the enzyme, then it can use the ATP. Okay. All right. Okay, let me go back. The question was asked, what is this PP molecule? Those are phosphates. So we popped a couple phosphates together. Yes, excellent. So you use energy or ATP by taking the time to make the next phone call. And then you end the call, which is like taking the phosphorus off and hanging up. Yeah. So that's exactly, that's a really good analogy. Thank you, Angel. Perfect. So it's kind of like taking the energy to make that call and then hanging up. And then the next person takes the energy to make the call and then hangs up. Okay, good. I love that you guys get my analogies and I'm not just like being a weirdo. Makes me feel a little better about myself. Okay, a couple of you said you're having a little bit of trouble with Wi-Fi, getting this, all that. I'll post this recording, I promise. Finally figured out how to do it. All right, so here's our response. We've received it, we've sent it, and then we can have a response. Now this response can be in a couple of different ways. We can regulate protein synthesis by turning on or off a gene. So when we make proteins, it's because a gene has been activated that says, I need this protein. Okay, so if we turn that off, we turn off making the protein. Okay, for example, if we don't need any more estrogen being produced, we tell it to stop and it stops making that protein. We can regulate activities of protein in the cytoplasm. So these can actually regulate how much protein is being produced because the signal can turn on or off the mechanisms of making protein. And I know it seems weird that we're talking about this now. This is going to make more sense in the next unit because the next unit is all about how we actually make the proteins. So remember that we're building and sometimes we look back and sometimes we're like getting ready for next units. All right, so here we see we've received it, we've sent our signal, and then this last molecule is going to actually trigger inside the nucleus to tell this, okay, it's time to read the DNA. We open it up, we create our mRNA, which then goes out into our cell and is made into a protein, okay? It can also be one that comes in and says, okay, stop. let go of the DNA. We don't need it anymore. So we can turn things on or we can turn things off. And the same thing happens in the cytoplasm. So let's say that this is an activity that's happening out here in our cytoplasm. Instead of going into the nucleus, it just goes over to that molecule and then says start or stop. All right. So some examples of problems that can happen. Diabetes, cholera, autoimmune disease, cancer, neurotoxins, poisons, pesticides, drugs. Let's start with diabetes. Diabetes is when sugar within the blood is not used properly within the cell. And that can be for multiple reasons. Okay, so there's insulin, which you guys are familiar with, is a signal that is sent in our blood to tell our cells, open the gate, let in the sugar. But if you don't make insulin, you don't open the gate. You don't let the sugar in. All right. We can also have type 2 diabetes, which is what's called insulin resistant diabetes usually, which means that even if there's insulin present, sometimes there is, sometimes there's not, the gate isn't receiving that signal. So it might get the signal and it's like, I don't know you. I'm not opening. We're not letting that in. Okay, so that's a problem. So it's a signal issue. All right. Now cholera. This is cholera. You can see here it has a tail. Anyone remember what we call that tail? Remember back to our cells unit? It helps it move. Oh, awesome. Look at my chat feed. Perfect. Flagella. Very good, guys. Now, this is a disease acquired by drinking contaminated water with feces. And a lot of times human, doesn't have to be cattle. We found that cholera can live within fields that cattle tend to pull up. We don't see cholera in the United States anymore. We've pretty much eradicated it because our drinking water is treated and we take care of the systems. It's tested regularly. We make sure that's not in our drinking water. But in other countries where we don't have that, they still have cholera happening. And it's a bacteria and it colonizes the lining of the small intestine and produces a toxin there. Now, that toxin modifies the G protein. So that protein that gets like shot out like the arrow or slingshot, it modifies that protein and it doesn't do its job. Excuse me. So it can't regulate the water and salt within the intestines properly. So now your intestines, if we can't regulate that and it's allowing the water to flow into the intestines, what do you think is going to happen to the person? If we put lots of water in the intestines? I'm telling you, you're going to be very unhappy for one. But what would that cause? Yeah, there we go. Okay, a couple of you. Diarrhea. Yeah, cholera causes profuse diarrhea, uncontrollable, and people actually can die from dehydration due to cholera. Now that sounds like a pretty almost funny, but just terrible way to die is death by diarrhea. But it has to do with just an imbalance of water and severe dehydration. Okay, but this is one that the toxin from it actually changes the reception in the cells. Viagra, you guys know what this is, okay? So Viagra, that little blue pill, is used as a treatment for erectile dysfunction. Now, what happens is it inhibits hydrolysis of this to this. Don't worry what that is, but just know that it means that it can't do this signal. It can't send the signal to make this happen. And what that means is there is a prolonged signal to relax smooth muscles in the artery. So our arteries are the blood vessels that go away from our heart. They go away and they have muscle within them that can contract, okay? And it can contract to help us move the blood, but it also can contract to limit where blood is being flowed to. If we are not digesting food in our stomach, if there's no food in there, we don't need the blood to go there as much. So we restrict the blood flow, we close it down so that there's less blood flow to the stomach. Okay. Now, what Viagra does is it causes the walls of your arteries to relax, meaning they're open more. All right. Well, that includes blood flow to the penis. The penis, the blood flow can change. And generally speaking, you don't need a lot of blood flow there most of the time. So they're generally pretty small and tight. So when we relax those arteries, they open up and it allows more blood flow in and doesn't restrict it. But this can be dangerous to the body if it's used frequently, because that means all those blood vessels open. And to be honest, what happens if you open up all your blood vessels and they're open for too long is all the blood like drains. And now your brain's not getting blood. But it also can cause damage to those artery walls. So individuals that have Viagra as a treatment usually only have a limited number per month, something like 4 per month, because it can cause damage to the system. Apoptosis. We talked about apoptosis before. It's self-suicide. I love that word. It's just so much fun. Apoptosis. Apoptosis is cell dismantling and digestion. So it receives a signal that says, there is a problem with this cell, okay? So a cell can become invaded by a virus or a bacteria. It can have damage from inside, such as cellular DNA damage from radiation, such as the sun, all kinds of different things. But it's a signal that that cell then has that says, there's something wrong. I need to be destroyed before I hurt anybody around me. So the cell will actually collapse in on itself and start being degraded, broken down, and recycled. And we also use it for development and maintenance. Okay. And there are studies being done now that this cell death may be part of what's involved in Parkinson's and Alzheimer's disease. Now, both of these diseases involve a degradation of the brain where cells are basically dying away. Okay. And when you have your brain cells dying away, that means that the memories aren't there. They're gone. That the ability for your body to regulate its actions, so movement, is also degraded. Okay. So they're studying that right now. And then when we interfere with apoptosis, so when a cell can't die when it's supposed to, that is believed and is being studied to see if it is a factor in the production of cancer within the body. All right, now this is a human white blood cell. This is another white blood cell, but this white blood cell is collapsing. It is being recycled. Now, doesn't necessarily mean that it's bad or something wrong with it. It could just mean that it's worn out and it can't do its job as well. Your red blood cells, they form, and then within 3 months, they are degraded. And you see these like blobs, they call them blebs. Okay, so these blebs, it's parts of the cell are collapsing, which forces other parts to kind of bleb out, and it creates this weird structure. Eventually it gets like just broken down and collapses. Now this to me is super cool. This is showing development of a mouse, the hands of a mouse, while it's developing it as an embryo. The fact that we can get these images is amazing. But if you look at the one on the far left, your hand started just like this, like a fin or a flipper, okay? No definition to it. And then the cells are programmed to die, creating the individual little, what would you call that? The spaces between your fingers, okay? Mice have 5 fingers just like us. Okay, so all of these cells die away, leaving us with our hand. And if it doesn't die away, then we wind up with things like webbed fingers or webbed toes. So if you've ever heard of or known someone who has, it happens more frequently in toes, but if they have webbed toes, it means that the skin will still be up. For some people, it's very short. Let's see, there we go. It might be really short and just kind of like, you know, little bit, but for others, they can have toes that are completely webbed all the way to the top. And it just means that when they were developing inside mom, this tissue did not undergo apoptosis like it was supposed to. Okay, pretty cool. Okay, your nervous system. Your nervous system relies on communication. It is the signaling system. And if I can't tell one neuron, the message to send to the next neuron, then the signal's dead and our nervous system isn't working. We don't get those signals out. That means that when you step on that Lego, your brain can't go, oh, that hurts, you idiot, move your foot. So these are super important. And I told you that our nervous system works mostly on electrical impulses, and that's correct. So this is going to be mostly an electrical impulse that goes down, but we can't make electricity jump. So we have to have a chemical that goes from one neuron to the next. So we have electrical, then chemical, then electrical. Okay, and we're going to look at that just a little bit. Now, a neuron, a cell from your nervous system, is just like every single cell we've ever looked at. So imagine that fried egg shaped cell. We're going to take that cell and we're just going to start pulling in weird shapes out of it. Okay. So we still have a nucleus, we have endoplasmic reticulum, we have all of that stuff. And then this would just be like as if the membrane was pulled into weird shapes. Okay. So it's just a normal cell. All right. Now we have what's called membrane potential. This is the difference in electrical charge across the cell membrane. Okay. So we can look at the membrane and put an electrode in there. We can actually see that it has a negative 70 millivolt charge of electricity, which to me is super cool to think that we have electricity, that we can actually measure electricity in our bodies. All right. So here is our neuron. There's our neuron. We're going to take a little piece, just look at the edge of it here. So here's our cell membrane. Here's proteins within our cell membrane. Here's inside the cell and here's outside the cell. All right, now we have our channels and we have things that can go in and out. Okay, and we use our sodium potassium pump like we talked about before. So our sodium potassium pump allows us to move the sodium and the potassium at different rates. So we're not moving one potassium for one sodium, we're moving them at different rates. Now, our resting potential is what it is at rest. It is polarized. That means that we have more sodium outside, more sodium outside, more potassium inside. And we have those gated channels, they stay closed. And a nerve impulse or a stimulus causes a change in that, which depolarizes. Okay, and it's all or nothing. Okay, we want to see the pictures. There we go. So if we take a look here, this is our membrane. We have, it's more negative outside, more positive inside. And we have a gate here. So you can see it's almost like if we had a ball on a string and we could stuff that up in there to close it. All right. So by opening this, we allow these to flow in. By closing it, they can't. All right, so if we start here with one, I don't know why it goes in this weird direction, but we start here at one. Here's outside the cell. We have a signal that removes this, opens our gate, and allows the flow. Okay, and if we do that, we can actually change where the positives and negatives are. So here we change that flow and it goes from being positive outside and it's going to switch it because they went through. And because it goes through, it switches it. We wind up with this voltage and it then triggers the next ones to do the same thing, allowing that voltage change and allowing that action potential to move along the neuron, sending the signal down the axon. Okay, I know that seems a little weird, but it allows this area, the signal part, to be sent because it flips and then flips back. Okay, flips and then flips back, allowing, see the blue part kind of moving along. Imagine like doing the wave in a stadium. That's how it moves down the axon. Now, once it gets to the end of the axon, okay, we call it the presynaptic, that's before the opening. This is our synapse. We have our presynaptic neuron here, and it is going to release neurotransmitters. So once the signal comes down here, we have the potassium that comes in through that channel. It triggers this to be released into that synapse. So this chemical, our ligand, then attaches here. So it attaches and opens it up, allowing the sodium and potassium on the next neuron to start switching spaces. Okay. Now, neurotransmitter is a type of ligand. It's released from the neuron into the synapse. It diffuses across it and binds to the receptor. Okay. It is then broken down by enzymes and taken back into the surrounding cells. So we release the ligand, and then we have enzymes in that space that start breaking it down. We have excitatory neurotransmitters that speed up the impulse, and inhibitory, which slow it down. That's like opening versus closing. So acetylcholine. Acetylcholine stimulates muscles, memory formation, learning. So think thousands of years ago, okay? People running away from something big and scary that wants to eat them. If that happens, your brain stimulates the muscles to be able to move. And it also stimulates the brain to form memories so we can learn, such as if I go here, I'm going to be chased by that big scary and it might kill me and eat me. Okay, so acetylcholine is definitely an evolutionary thing that helps us survive. We need it. Right now, maybe not to that extreme, but we still have things like acetylcholine would be released when you go down a dark alley, especially for women if you walk down a dark alley, if you're walking to your car in a dark parking lot. Any of those situations that we feel danger, we're going to remember and our body is going to respond to that. Okay. Now, nicotine binds to those receptors, okay, and it inhibits it, slows things down, but it increases heart rate. Okay. So nicotine, that's why it becomes addictive, is it kind of makes you feel calm. Now, sarin is nasty, nasty stuff. Sarin is a gas. It's a nerve agent that breaks down acetylcholine. Okay. It inhibits the breakdown of acetylcholine. That acetylcholine builds up and it causes paralysis, meaning you can't move. And that includes your lungs can't move, your heart can't move, and you die. It works very quickly. So it does all this very quickly, but remember that it does take 3 to 4 minutes for you to die once everything stops. So it stops everything and then you lay there and die. This is nasty, nasty stuff. It's used as a weapon. Okay, bad news. Next we have Botox. So Botox to me is a total trip. This is botulinum toxin. This is the most toxic substance known to man. We haven't found anything more toxic than botulism. Okay. This blocks the receptors on the muscles and prevents muscle contraction. If you get botulism poisoning, it interferes with how the muscles work. Now, Botox is used in the beauty industry. It's also used in the medical industry because it stops muscles from contracting. So in the beauty industry, they'll inject it in the face because if those muscles can't contract, that means that things can't wrinkle up because they're, so it makes everything nice and smooth because they can't contract. It's also used in medicine though. So the muscles on the outside of the scalp are connected to the neurons in the back, okay? And when those muscles pull, it can actually cause headaches. They will inject small amounts within the scalp and within the forehead to reduce that from happening. So that helps a lot of migraine sufferers. I did it for about 3 years where they would inject, it was about 30 different shots into my head and neck, okay? And it works pretty well. People who have severe muscle spasms, they'll I was reading an article about a gentleman that had severe muscle spasms in his neck and it would cause his neck to twitch and his head to spasm. So they routinely inject botulism into that muscle, which reduces the muscle from contracting. So now he doesn't have those. Okay. Now ligands are generally used up. We've broken down by an enzyme. So once the ligand is released, it gets broken down by an enzyme so that we have to produce more to continue signals. That was a good question, Fiona. Thank you. All right, now epinephrine. Okay, that's adrenaline. That's part of our fight or flight. You guys know how that works. You get startled and your heart starts racing and your muscles they almost will feel like, I don't know, twitchy or kind of like tingly. That's because they're getting ready to be able to run. Now, this fight or flight system does not have to be an actual danger. You can just feel like you're in danger. If you've ever seen any of the Halloween videos where the person jumps out and they get punched in the face because the guy freaked out, that's the fight or flight system. But it also happens if you're riding a roller coaster, you know, when you're going up, that like, you get to the top and your heart's like, nothing's ever happened. You're not in danger. You know you're not in danger. Well, maybe a little bit of danger. Am I in danger? I don't know, but your body freaks out. Definitely not in any danger when you're watching a horror movie and something jumps out and you jump and you scream. Okay, so we release these signals when there is perceived danger to help us stay safe. Dopamine, this one is our reward. This is the pleasure. This is kind of gives you that high. Dopamine is released in mass quantities by your brain when you are infatuated with someone. Okay, you're around them and you just feel good to be around them. That's because you're releasing all kinds of dopamine. If you're an athlete and you're working out and you hit the point where you're like, yeah, it hurts, but I feel good. That's the dopamine that's been released. So your body hits the point where it's like, this sucks, this hurts, this isn't fun. Okay, I'm gonna give you a little bit of dopamine just to make things okay. This is also what is mimicked with a lot of drugs. And then your brain is like, wait, I want more of that. Wait, I want more of that. And so you keep feeding it with those artificial drugs. A loss of dopamine, meaning you don't produce dopamine, can lead to Parkinson's disease. Okay, several other things as well. Now, serotonin, this is well-being and happiness. This is the I'm okay. It's not the like, yay, but it's the, all right, we're good, it's chill. And people who have depression and anxiety generally do not have enough serotonin within the synapse. So it's released, it does its job, and then it's broken down really quickly. Okay, so that's a problem. What happens then is with medications, so things like, I think I have it here. I didn't have it here, but things like Prozac, Xanax, Cymbalta, Effexor, I mean, all those different anxiety and depression medications, what they do is they make the enzymes that break down the serotonin in the synapse, okay? Those enzymes are like, it makes it so they don't work so fast. That way the serotonin can remain in there a little longer. Now, it's normal for the serotonin to be in there for a while, but with people with anxiety and depression, they break it down too fast. So it's called an SSRI, selective serotonin reuptake inhibitor. So we're not taking it back in as fast. So people who have anxiety or depression, usually what these medications, once they figure out the correct dosing and all of that, they just put their brain back into a normal state. So that's really cool. For some reason in the United States, we have a huge stigma on taking medications for anxiety or depression, but they're no different than taking insulin if you have diabetes. Your body's not producing what it's supposed to, so we're gonna help it out. Now, is this a psychological or genetic condition? It is actually, there's a huge component of both. So genetically, there are people who, like you'll notice that anxiety or depression runs in families. That's because genetically, your body doesn't know to make enough of the serotonin. And then psychologically, when we're under stress, we actually release chemicals that will break down other proteins. And it's cortisol. It causes us to gain weight, which a lot of us have done during quarantine, and it causes chemical neurotransmitters to be broken down, which affect our memory, which can affect our stress levels, all those things. Okay. Now, the question was asked, is depression a physical illness rather than a mental one? Well, it's kind of like saying, is diabetes a physical illness or an endocrine one? So it is a physical illness that causes a mental, what we see as a mental change. Okay, so it is something that's physically happening within the body. That's why we can give medication to help it. But then there's also external factors such as behaviors and all of that, that mean that we can adjust how we act and react that can help our body manage those chemicals, if that makes sense. And a lot of what we call mental illnesses, we treat them with medication, and that they're all kind of the same way. It means that either something's being produced too much or not enough, and we're balancing that. That's what most mental illnesses are. Now, GABA. GABA is an inhibitory neurotransmitter, which means it slows all the other things down. It can be affected by alcohol. Alcohol is a depressant, so it makes everything in your body like slow down. It reduces your reaction time. It reduces your inhibitions. It just basically slows everything down. Now, anytime somebody's had enough alcohol to inhibit enough GABA, then they are actually risking death. If somebody passes out because they drank alcohol, they are risking death at that point because they've slowed enough of their body down that their brain is basically like, nope, I'm checking out for a bit. And you can actually wind up slowing down your heart and that to the point of death. Okay, so excess alcohol is very bad. LSD or mescaline binds where the serotonin and dopamine should be, and it causes hallucinations. Prozac, let's see, dopamine. Is dopamine released when you're hurt? Generally, no. When you're hurt, you're going to release other things that will basically block that. The ephedrine, epinephrine, norepinephrine. There we go. Too many chemicals in my brain right now. Names of chemicals. So our endorphins, those things will block pain. Now the dopamine is released for athletes, not because they're in to block the pain, but kind of a way to balance what's going on in the body. Okay, they call it a runner's high. Again, Prozac affects serotonin uptake. Morphine and heroin bind to the endorphin receptors, which decreases pain. Morphine and heroin are common painkillers that are used in hospitals. The problem is when it's used, it shouldn't be. Viagra, we already talked about. Alzheimer's disease, it's develops senile plaques and shrinkage of the brain tissue. Okay, so again, shrinkage of the brain tissue means death to cells. So cells are dying. Okay, so all of these things that we're talking about with the nervous system, these are all based on signals. And without our nervous system functioning properly, our body can't function properly. Our nervous system is what tells our heart to beat. It's how we get our lungs to do their thing, all of that. So signals, we need signals for things like the endocrine system, but we need signals for communication with the nervous system. So we have two sides. Now the nervous system is going to send quick, rapid signals. I stepped on a Lego, move. Whereas endocrine is going to send long-lasting signals like, okay, it's puberty, time to change the body. We don't want it to, go backwards. We don't want it to be all of a sudden. So puberty happening in like 10 seconds would be so incredibly painful and like weird. And if it went backwards, and so we want that to be long-lasting and slow. Whereas our nervous system is a quick, very quick signals and communication within the body. Okay. And that is a ton of information in just about an hour. Do you guys have questions? Okay, I'm not seeing anybody typing frantically yet. If you have questions, please feel free to keep posting them and hang on. But otherwise, I want you guys to take these notes. I want you to take a look at handout 4, okay? And we're gonna be doing notes on 4/2 on Monday, Tuesday for you guys. And that is gonna start with the cell division. So at this point, we've covered half a unit. Yes, Dylan, right where I was going. So this is half the material up to right here. Okay, so it's just, we have our signal, which is reception, transduction, and response. We have different types of signaling. We have what they do and why it's important. All right, well, with that, I guess I will see you guys, let's see, Tuesday. I'll see you guys on Tuesday, and I'll make sure to get this posted. Bye, guys.